the different kinds of immunity. So we talked about passive and active immunity, and then artificial versus natural. So today, mostly what we're going to be talking about in our map is this piece right here. So last time we talked about the non-specific defenses, you know, all the physical defenses, the phagocytes, interferon, all those. So that falls into this category. So today, we're going to talk about the other branch, which is the specific or the acquired branch. So we're going to talk about cell-mediated immunity, that's T cells, and then we're going to talk about humoral immunity, which is the antibody system. But first, just some key words about immunity. So in this top box here are things the immune system has to be able to do in order to be effective. Regardless of how it does it, it needs to have these four characteristics. So one is specificity. What we mean by that is the immune system should respond to different pathogens differently, because different pathogens are different. They have different weaknesses. They have different characteristics. So to have the best possible immune system, the immune system would respond specifically to specific pathogens. So we have specificity. The way this is accomplished is T cells and B cells, they don't work as individuals. Like the immune system doesn't work with individual T cells and B cells. Instead, they work in huge numbers. So you can think of it a little bit like an ant colony. You know, a single ant can't do very much. But because there are huge numbers of ants all doing different things, the hive can accomplish all kinds of impressive things. And in many ways, the acquired immune system is like that. Huge numbers of cells make it possible to accomplish these four characteristics. So in terms of specificity, each T cell and each B cell is looking for only one little part of would-be pathogen. So T cells and B cells are, each one is looking for just one thing. So it makes the system specific. Well, that's fine, but we need to look for lots of different things, not just one thing. So enter versatility. The immune system has to be able to deal with a wide variety of pathogens. So if each T cell and B cell is looking for just one tiny piece of a pathogen, well, how do we get a response to a wide variety of pathogens? By having huge numbers of T cells and B cells. So, we have millions or billions even of T cells and B cells, each looking for a different thing. So we have both specificity and then versatility because of this large population of different cells. All right, what about these other two? Memory. Ideally, we would like the immune system to remember its past events so that if a pathogen is encountered for us the second time in a lifetime, the immune system will already know how to attack it. And we call that immunologic memory. And the way this comes about, as we'll see later on today, is anytime the immune system is anytime the acquired immune system is activated, some cells don't participate in the attack. They just are made so the system will remember. So they're called memory cells. So we'll see that in an immune response, some of the cells simply remember for next time. So we have our memory. And then finally, and very importantly, is tolerance. We don't want our immune system attacking normal, healthy cells. So we have to have what's called tolerance. So the immune system will attack something that doesn't belong, but never attack something that does belong, that is normal. So one of the ways that this is accomplished is in the thymus gland. In the thymus gland, the T cells sort of learn the difference between cells that are part of the body and healthy and normal, and non-self cells, things that don't belong. So we have these four characteristics: specificity, versatility, memory, and then tolerance. And we'll see how those sort of play in as we look at the immune response in some detail today. All right. So as an overview of the immune response, the first, before our picture here, so if we made a box here, would be the non-specific defenses. So that's the first thing that's going to happen. But once they've started to do their thing, we start bumping into the specific defenses. And we're going to see that there's this thing called antigen presentation, that the phagocytes of the innate immune system, they present a potential infection to the specific system so that it can start its response. So once the acquired immune system is triggered, we get sort of a two-pronged approach. We get a cell-mediated approach, called cell-mediated because it's cell-to-cell, so to speak. So we have the phagocytes get activated, T cells get activated, and then there's a physical and chemical attack on whatever the pathogen is. So that's one branch, that's the cell-mediated branch there. And then at the same time, the antibody system is triggered because antibodies are, in many ways, our most effective weapon against pathogens. Because once antibodies are formed, the pathogens really don't stand a chance in the body. So eventually, while the cell-mediated immunity is happening, the antibodies are getting produced as well to finalize the destruction of those antigens. All right. So first up, what's an antigen? In some ways, I think this is one of the hardest pieces of the immune system to understand, which is why I came up with this big long slide that's not in your book. But okay, so the acquired immune system, it uses this system of molecular identification called an antigen. Why? Because it's easy for us to sort of think about cells like they're little people, but the reality is they can't see and they can't hear because they don't have eyes and they don't have ears, right? But yet, in order to accomplish their task, they kind of need to do those things. And an immune system cell needs to be able to see a pathogen in order to do anything about it. It needs to be able to know where there's something going on. So the system that evolved is this antigen system. So an antigen is simply a piece of a thing. So if you think about a bacteria, it's obviously very simple. But on the inside, there's all kinds of stuff inside a bacteria, just like there is inside our own cells. There's DNA, there's proteins, there's carbohydrates, there's lipids, there's all kinds of stuff. So an antigen is a little piece of something. So, for example, let's say that this bacteria has this piece here is part of a protein that makes up its mitochondria, let's say. Well, if this bacteria is destroyed, boom, a macrophage destroys it. What you end up with is lots of pieces, right? Because, you can't technically make things disappear. You can only change what they are. So in this collection of pieces, in the shrapnel, so to speak, from the death of this bacteria, that little piece of that protein is still there, right? So this would be an example of an antigen, a little piece of a larger thing. Now, why is that important? Well, because particularly living things like bacteria and viruses and things like that, viruses kind of alive anyway, there's so much variability in in what bacteria are made of, is identifying a piece of the thing is enough to identify the whole thing. So, think about some complicated machine, like your car. An expert mechanic could tell you the year, make, and model of your car from a single part of your car, right? Without even necessarily seeing your car ever. It's another example of an antigen. So an antigen is just a piece of a thing that is used for identification. So as another example, if a protein or a bacteria has a protein and it has this pattern of amino acids, if we see that pattern somewhere else, we can kind of assume that that is part of that bacteria that has that pattern. So that is an antigen, is a little piece of a thing. Now, it can be a piece of just about anything. So it can be a part of a protein, a lipid, a carbohydrate. It can even be a part of something synthetic, like plastic or mesh or wire. All of those things can be what we call antigenic. In other words, they have enough variability in their pieces that the immune system can identify those parts. Because what we're going to see is this antigen identification is how the whole acquired immune system works. It's sort of the way the cells see what they're looking for. You know, they find what they're looking for by looking for just one little piece of it. All right, so that's how they see. And in some ways, you can think of these immune system cells as being able to hear, too, because they can follow a signal. You know, if somebody, shouts your name from out of the corner of the room, what do? You turn to look and then maybe walk towards them. Well, the immune system cells can do that same trick, only it isn't with sound, it's with chemical signals. So we have these, two different systems for sort of seeing and hearing. All right. Okay, so antigens work very well for this identification. However, they don't work as well when they're on the inside of cells. The T cells and the B cells that make up the acquired immune system, they are in fact cells, so they can't go inside another cell and look around, because cells just don't do that. There's no mechanism for that. So if the immune system is going to identify an antigen, in other words, a pathogen, that's on the inside of a cell, we're going to have a system for that because, cells can't go inside other cells. So that system is called the MHC system, the MHC. And that's the complex word there is it's the major histocompatibility complex. But all I want you to know is MHC. And there are two types of these MHC molecules. Okay, so if there's a foreign antigen, a piece of something that doesn't belong, if there's a foreign antigen inside a cell, what the cell does is it takes that antigen and it packages it into one of these MHC molecules and then displays it on its surface. So it's sort of like hanging a sign on the outside of your house, where the house is the cell. So if the cell finds something that doesn't belong, it puts it on a poster, so to speak, and hangs that poster outside the house. And the poster is the MHC complex. The antigen is the little piece of the thing that you found that didn't belong. And MHC molecules are sort of special in the body because they are profoundly different from person to person. So, it's only in identical twins that you can find two people with the same major MHC molecules, major histocompatibility complexes. So they're highly variable. And without getting into all the details, that variability means that they're an excellent way for the immune system to tell your cells apart from some foreign cells, right? So the MHC, so there's two types. There's class 1, and I want you to know this bit, and there's class 2. Class 1 are found on all nucleated cells. So every cell in your body that doesn't have a nucleus, or that has a nucleus. So what is that leaving out? The red blood cells. Red blood cells don't have class 1 MHCs, but they're the only exception. All other cells have these class 1s. Class 2 MHCs are only found on what are called antigen-presenting cells. In other words, immune cells. And the fact that there's two classes does kind of matter to how the system works. So we have class 1s found on all cells, class 2 found only on antigen-presenting cells, which are always immune system cells. So let's look at the class 1 system. All right, so this is any old cell right here. So this isn't necessarily an immune system cell. This could be a skin cell, a digestive tract cell, even a nerve cell. So this cell has been infected with a virus, right? So here's the viral DNA coming in, going to get replicated. Well, the systems never work perfectly for viral infection. So some of this virus ends up as little pieces. Those little pieces are identified by the cell as something that doesn't belong, right? So, just like we talked about the poster on the outside of the house, that those pieces, those antigens, are packaged into a major histocompatibility complex, so a class 1 MHC here, and then they go to the surface where it's displayed. So inside, in purple here is the MHC molecule, and in red is the antigen. All right? So in the absence of viral infection, in the absence of infection here, there are still MHC molecules on the cell surface, but they don't have any antigens inside. So it's like you have an empty poster, where once there is an antigen present, it's a sign to the immune system. The cell is essentially saying, I'm infected, and this is what I'm infected by, is this antigen. So then once that MHC molecule with an antigen is present, now the immune system can do its thing and can identify this cell as infected. All right, so that's the class 1 system. In the class 2 system, it's pretty much the same general effect, but the cause is different. So in the class 1, how did this antigen end up in here? Because this virus entered the cell, created this antigen, which ended up in that package, right? In the class 2, it isn't an infection that triggered it, it's phagocytosis. So let's say that this is a macrophage or a neutrophil. So it's gobbled up a bacteria here, and it's swallowed it in. The lysosome contains powerful digestive enzymes. So we turn an intact bacteria into a bunch of little pieces of what once was a bacteria, right? Well, all of those pieces are potential antigens. So what the phagocytic cell then does is it takes one of those antigens, packages it into a class 2 MHC, and then puts it on its surface. So in the end, we end up with the same thing. For the class 1, we have an antigen in an MHC complex. For a class 2, we have an antigen with an MHC complex. The difference is how it got there. This cell is not infected. It just killed something that it wants the immune system to know about. So it displays that on its surface. We call this antigen presentation. And the phagocytic cells and the other cells of the immune system are the only cells that can really do this. And it's important because these class 2 MHCs are what trigger the antibody response or play a role in the antibody response. So we have class 1s on all nucleated cells. Class 2 is part of that antigen presentation system. All right. Okay. So then what comes next? Every T cell and every B cell has just one antigen that it's looking for, right? So in order for that T cell or B cell to become activated and trigger an immune response, it has to find its antigen. And we call that process antigen recognition. All right. So here's a class 2, here is a class 1 MHC, could really be either. So we've got an antigen in an MHC complex, right? So along comes a T cell, and it sort of backs up against this cell, and it checks to see if the antigen in that MHC matches the one that it's looking for. If it's a match, it'll activate. If it's not a match, it won't do anything. It'll move on. It'll go do something else. So because there are huge numbers of T cells, each with a different thing they're looking for, in a new infection, at least one of those T cells is going to find its antigen that it's looking for. And once that happens, then the inactive T cell starts the process of activation. So it's primed, so to speak. Now, there are two different kinds of T cells that we care about. There are TC cells and TH cells. TC, that's cytotoxic. These are T cells that attack other cells. H is helper, right? And there is sort of a code associated with these. All right, so CD8 and CD4. Okay. All lymphocytes kind of look the same under the microscope, right? So we have to have ways of telling them apart. And one of the ways is to look at what's on their cell surface. So cytotoxic T cells are often called CD8 cells because that's a marker on their surface. T helper cells are called CD4 cells. Now, why on earth am I getting into this much detail about this little thing? Because of HIV. HIV affects the T helper cells. So when in taking care of patients with HIV and to understand that illness, you have to know that this is what HIV attacks, is the CD4 T helper cells. So that's the only reason I'm getting into that detail. And the last connection I want to make about this sort of code system is CD8 goes with class 1 and CD4 goes with class 2. So the cytotoxic T cells, the CD8 lymphocytes, they interact with class 1 MHCs. The T helper cells, the CD4 lymphocytes, interact with class 2 MHCs. So a little trick I have is, so if 1 goes with 8 and 2 goes with 4, you can think of 2 times 4 is 8 and 1 times 8 is 8. Does that kind of make sense? So it's just a little trick. So how do you remember that 2 goes with 4 and 1 goes with 8? Because when you multiply them together, they both have to make 8. Little helper there. All right. Yes?
On the homework, there's 2 questions about what on MHC1, what cell that activates.
Mm-hmm.
So would that be the T or
Cytotoxic T cells, CD8
okay,
cells.
and then the answer for the other one would be T helper.
Yes, the class 2s activate the T helper cells. Yep, you got it. The class 2s also activate the B cells as well, which we'll see later on. All right. So all that, some just little details that you might have to know for some other test at some other point in your life. But this is antigen recognition. So the T cell finds the antigen it's looking for in a class 1 or a class 2, depending on what kind of T cell it is, and then it's activated. So that's this piece. And then we have what's called co-stimulation, which is a sort of complex how it works. But we don't want the immune system to respond unnecessarily because it's a killer. I mean, it's going to kill cells to solve like a viral infection or a mutation. So the system has kind of a safety on it, and that is once a T cell finds its antigen, like here, it's primed but not active yet. In order to become active, it has to be co-stimulated. So essentially, it has to find another cell with the same antigen to turn it on, so to speak. And then once it turns on, now this T cell's life is about to change because now it's become important. Instead of just looking for its antigen, now it's part of an immune response. So the first thing that T cells and B cells do when they're activated is they start dividing. So they make many copies of themselves. And those copies are not all the same. So most of the copies are going to become part of the attack force. So they're going to become these cytotoxic T cells that are going to seek out that antigen and destroy any cell that has that antigen on its surface, right? So that's most of the cells. So that's going to damage or it's going to destroy the infected or mutated cells of concern. But then some of the copies don't do that. Some become memory cells. In other words, they return to an inactive state and they sort of go away. They are still circulating in the body's system, but they're not participating in this immune response. So because there are these memory cells, the next time we encounter that antigen, the body's response can be much faster and much larger. So some become memory cells, and then some become suppressor cells. Suppressor cells turn the immune system off, or turn this particular immune system or immune response down. Important because everything has to have an off switch. If you're going to turn it on, you have to be able to turn it off. So the suppressor T cells develop later in the infection. So by the time the infection is about cleared, the suppressor T cell numbers start to go up and turn off the original immune response so the body can sort of get back to normal. All right. So the way that the cytotoxic T cells, deal their death to infected cells. It's the same way as the NK cells did that we talked about last time. So those perforins are released. T cells, cytotoxic T cells can also trigger a cell to kill itself. So it can send a signal that will make the cell sort of involute and pop all by itself. And then another is there's a lymphotoxin there too. So there are many ways that the cytotoxic T cells actually, deal out the destruction there. All right, so if the helper T cells destroy infected or mutated cells, what do the helper T cells do? Well, they do just what their name implies. They help the rest of the immune system do a better job. So in order to activate a helper T cell, you need to have a class 2 MHC, which means you're talking about an antigen-presenting cell. So you're talking about an immune system cell. A neutrophil, a macrophage, another lymphocyte, some other cell like that. And the process goes the same. First there's activation, then there's co-stimulation, and then that T cell is activated. When T cells activate, the first thing they do is start dividing. So some of the cells become memory T helper cells, and then some of the cells become activated. And their big role is they produce chemical messengers, namely cytokines. And cytokines are a big class of sort of like hormones that help to control and stimulate the immune system to do its thing. T helper cells, as we'll see in a minute, also play a role in activating B cells to become plasma cells and start making antibodies. All right, let's do a question or two. When an antigen is bound to a class 1 MHC, it can stimulate what kind of cell? Did you get the question? Yeah? Anybody?
I got it.
Oh, must have worked. I'm getting answers through. So. Are you jumping there, last person? No, I've got 5 of you and I only have 4 answers. All right. So class 1 goes with D, cytotoxic T cells, which most of you got. And do make that association. So CD8, class 1, cytotoxic T cells. If this question had said a class 2 MHC molecule, it can stimulate a CD4 T helper cell, right? Okay. Class 2 MHC proteins are present in the plasma membrane only when? Or only which of those? Because even though I just said when, that is not necessarily the correct answer. All right, so the best answer here is B. So class 2 MHC proteins are associated with antigen-presenting cells, so things like neutrophils and macrophages, as well as lymphocytes. The lymphocytes themselves also have class 2 MHCs. So let's look at some of those others. E, during infections. The class 2 MHC is present on the membrane all the time. It's just empty. It's only during infections that the class 2 has an antigen inside. So don't sort of mix those up. All right. All right, so that's T cells. You know, T cells are activated by these or by these MHC molecules. They become either cytotoxic T cells that do what NK cells do naturally, or they become helper cells that assist the immune system in what it's doing. So what about B cells? Well, thankfully, there's a lot of similarities. You know, a B cell, just like a T cell, has one particular antigen that it's looking for all the time. Only for B cells, it doesn't have to find that antigen in one of these MHC molecules attached to a cell. A B cell is sort of looking everywhere for an antigen. So on cell surfaces, in between cells, in the interstitial fluid, the B cell is looking for that antigen everywhere. So when it finds that antigen it's looking for, that antigen binds to it. So our red dot here is the antigen. Here's the B cell with its feelers out, so to speak. It's actually a form of antibody that it uses to do that. And then it finds a match. All right. So once that happens, we call the B cell sensitized. Now it isn't activated, so it hasn't started dividing and changing yet, but it has found what it is looking for. So then the next stage is activation. So in order for a B cell to go from stimulated to activated, it has to interact with a helper T cell. Okay? So first the B cell finds what it's looking for, and then it toddles off to find a helper T cell that has found that same thing that it's looking for. So it's sort of a handshake. You know, the B cell says, this is what I found. The T helper cell says, or shows, yes, that's a bad thing. You know, let's get rid of it if we want to destroy that antigen. So once that activation or co-stimulation happens, then the B cell is activated. Okay? So in order to get an active B cell, we have to have an active helper T cell first. So you can see that, you know, like HIV, which attacks helper T cells, if we knock helper T cells out, it means the whole B cell system doesn't work because there's no helper T cells to turn the B cells on. So it's one of the reasons why that disease is particularly nasty. All right, but then, just like T cells, once a B cell is activated, first thing it does, divide. So we have this, what they call it clonal expansion, where many copies of that original cell are made. Some of them become memory cells. I'm going to deal with the next time we encounter this antigen. And then the rest become active B cells. Now, when a B cell gets ready to do its thing, which is to produce antibody, it does change in appearance pretty dramatically. It goes from a little tiny cell with a big nucleus and very little cytoplasm to kind of a monster of a cell with huge amounts of endoplasmic reticulum. So those are called plasma cells, and they're antibody factories. So B cells are stimulated or sensitized here when they find their antigen. They're activated by the helper T cells, and then they expand their population and begin producing antibodies. So they're very efficient. So like your book says, a plasma cell can produce 100 million antibody molecules an hour. So we're talking about a very efficient little factory. Yes?
Okay, so for one of the questions on the homework, it says it's primary activated. Would that be the antigen or the helper T cell?
What does the question say?
It says B cells are primarily activated by the activities
Of helper T cells. Yeah. Because sensitization happens first, but activation happens with the helper T cells. All right. All right, so what's an antibody? Well, it's a molecule that looks a little bit like this. And it has sort of two ends to it. And yes, it really is very Y-shaped like this. The top part of the Y, so here and here, that's where the antigen attaches. The top half of this molecule is highly variable. In other words, you have circulating in your bloodstream right now, probably 100 million different types of antibodies. And how is that possible? It's because the top ends are different. So each one of those antibodies has a little bit different top end so that it can connect to different kinds of molecules. Whereas the bottom end of the Y, this end is always the same. So of those 100 million different antibodies, the tail, the bottom of the Y of each one of those is identical. So we have a different region at the top, and we have a same region at the bottom. So this allows this molecule to behave as a kind of handle. You know, if the top, you've got a whole toolbox full of antibodies, each one is different. Well, you can pick one and attach it to something, and pick a different one and attach it to something else, but the handle, the part sticking back out at you, is the same. So that's called opsonization. So the different end attaches to the antigen, and then the same end sticks out so that the immune system can use it as a handle to grab hold of things. And there's a great picture of that here in a minute. Okay. So antibodies, when they bind to their antigen, they sort of stick together, and we call that an antigen-antibody complex, right? And it's these complexes can then be attacked by the immune system. So the antibody is attacked, but more importantly, the antigen is attacked. Also, when this happens, let's say this is a toxin, you know, like botulinum toxin. When the antibody attaches to it, usually the toxin is neutralized because it can't do its thing if it's got this giant antibody molecule attached to it. So chemically it becomes inert. And we're not going to talk about the whole partial antigen hapten thing because it doesn't come up. All right. So this is what makes antibodies useful to the immune system. You know, let's say that this giant thing here is a bacteria, and it has all of these different antigens that are part of it. Well, once you've developed antibodies to these antigens, this is what the bacteria ends up looking like to the immune system. So we have all these antibodies stuck to this thing, and every single one of these bottom end of the Ys, each one is a handle for the immune system. So it means that a neutrophil can come along and very easily grab hold of this and pull it inside and destroy it, because now it's got all of these handles. So that's one way to think of it, is as handles. Another way is as labels. If this is cruising by in the bloodstream, let's say in the spleen, and it has no antibodies attached to it, the immune system might not notice, you know? But because this has antibodies attached to it, it's like a giant red flag to every immune system cell in the body that this is something that needs destroying. So it also flags or labels stuff as it passes through the body so that the destruction occurs much more easily. All right. So antibodies come in 5 classes. If you look at the designs here, you can see they're all Y-shaped, right? But for M and A, we have put the Ys together in various patterns, okay? So just like I did with blood cells, I'm going to give you a sort of claim to fame for each one of these, because I do want you to know the 5 classes. So it's G, E, D, M, and A. All right, so the first one, IgG. Most numerous and diverse. Most of the antibodies circulating in your body right now are IgG antibodies. And IgG, they can be active against viruses, bacteria, toxins, just about anything. So most numerous and most diverse. All right. IgE looks almost identical to IgG and D. The differences are not important. What's important is IgE comes up with allergies. So people who are allergic, they have higher than usual circulating IgE levels. So I want you to associate IgE with allergies. All right. IgD, their claim to fame is they're actually found on the surface of B cells. They make up part of the feeler apparatus of the B cells. So if we go back in our pictures, so see, here's our inactive B cells. These are IgDs right here, these Y shapes. So they're sort of stuck on the surface of B cells, and they play a role then in sensitizing the B cells. So that's IgD. So it's part of that recognition system. Okay. IgM, not only is it the prettiest, you know, it has this sort of snowflake appearance where the ends of the Y are all sort of stuck together, and there's 5 of them that make it, that's kind of snowflake pattern. But its bigger claim to fame is it's the first antibody to be produced. So in a new infection, you've just encountered a virus you've never encountered before, the first antibody that you can see appear in the bloodstream is IgM. And you can imagine with all of these pieces sticking out like this is going to be a very vigorous antibody, right? Because it's every single one of these pieces sticking out is looking for an antigen. So it's very aggressive is a way to think of it. And then another thing is IgMs have a tendency to make things stick together. Because let's say, like red blood cells, you did blood typing. IgM, if you had a red blood cell attached here and a red blood cell attached here, what's going to happen? Well, they're going to be stuck together, right? Because the IgM is making a bridge. So they have a tendency to make things stick together. All right. And then IgA, commonly you'll hear this referred to as secretory IgA. And the reason for that is it's one of the only antibodies that is secreted into our other bodily fluids. So, saliva, urine, the reproductive tract, all of those places have IgA. And essentially, it's two IgGs stuck together at their ends. So they're sort of bound together. So we find the claim to fame there is IgA is found in secretions. We call it secretory IgA, yeah.
Are you ever going to ask us which one is the first on the site of infection? Because if you are, you said G was the most abundant and most diverse, which would make me think that you would find that on the infection. But then you said M is the first antibody to be produced.
M it would depend on if the infection was one you had seen before or not.
So if you were to ask a question like that, what answer are you looking for? Or would
I would
you?
think IgM
Okay.
because it's the first. Yeah,
Okay.
that's yeah. Is there a question like that in the homework? Or?
No, I'm just, after your test last time, I'm trying to think now.
Oh. All right, so, and we were just talking about this, how the primary and secondary responses are different. Okay, so your immune system doesn't respond the same way the first time you encounter a pathogen as it does the second time. So we call that the primary and secondary response. So, okay, so here's time in weeks, and this is antibody concentration, right? So somebody sneezes on you, and now you have a virus in your upper respiratory tract, right? So you're sick here for the 4 or 5 days. Somewhere about the time that you're starting to feel better, so at about day 5, let's say, now antibodies to that new virus are being produced. So the first kind of antibody we see is this IgM, which is right here. So the IgM level is going up, up, up. Following along behind that, about 4 or 5 days or 3 days, is IgG. So this is why we say that IgM is first on the scene, because in a new infection, first we see IgM, then we see IgG. But what we see is the antibody level goes up, and then the antibody level goes down. Now notice, it doesn't go back down to 0, right? But it does go down to almost 0. And IgG also eventually almost goes to down to 0 too. So over a month, we get down below there. So that's a primary response. There's an antibody production, but it wanes, it disappears quite quickly after the infection. All right, well, what happens if that same person sneezes on you again, and you get the same viral, you cleared the first viral infection, and 6 months later, you encounter that virus again. Now the response is different. Because the B cells created memory B cells, that antigen is very quickly identified, and the response is very quick to happen. So in the second exposure, or secondary response, we have a little bit of a different pattern. We get IgG and IgM sort of appear at the same time. And while the IgM follows the same pattern, if you compare this curve to this curve, they're almost the same, right? That pattern stays the same to fight off the acute infection, but the IgG level goes way up and sort of stays up. This is why there are booster shots. Why do we have to get a vaccine again to make this happen? It's the secondary response, particularly for diseases that are not in our communities anymore. Because, You got your measles vaccine, and this was the pattern, right? And then, 10 years later, you've never encountered the measles virus because measles is all but dead in America, right? So you never really got the secondary response. So then at 12, we hit you with the measles vaccine again so that we can get your IgG levels up very high so that if you ever do encounter measles, you're not even going to know you did, right? So we have primary and secondary response. So it plays a role in vaccines, but it also plays a role in every virus that we're exposed to again. So the first response is limited. The second response, when I think of it, is the second time the body sees a thing, man, it's going to make sure that that thing can never do any harm. So it's going to make a ton of antibody. You're going to get a lot of antibody levels real high, so there won't be any effect if you encounter it again. All right. So what do these antibodies do? Well, we talked about this one already, neutralization. Having a big old antibody molecule hanging off of you is probably going to interfere with what you're doing, whether you're a toxin, a virus, or a bacteria. So there's that. Having that big molecule prevents that thing from doing what it normally does. Prevention of adhesion. A bacteria, in order to invade the tissues, it has to be able to hold on to something, right? And if it's got antibodies all over it, it's not going to be able to do that because the antibodies are going to be in the way. The antibody system activates the complement system that we talked about last time. The complement system is a biochemical attack on bacteria. Anything that has antibodies attached is going to get attacked by every part of the immune system. So by complement, the antibodies cause opsonization. This is the handle effect, okay, that the phagocytes can now grab hold of this antigen or this bacteria because it's got handles all over it. Antibodies attract phagocytes as well. We've learned that they can do chemotaxis. They can follow a chemical signal. Antibodies are one of those chemical signals, or at least they trigger them, so that the phagocytes can find them. Antibodies stimulate inflammation, which we talked about last time. Inflammation is that common response of the body to injury. And then finally, antibodies can make things clump together. Now, how is that a good thing? If antigens are all stuck together, it means they're all in a very small space. They're in less space, so they can do less damage because they're around less cells. So we have that's called precipitation or agglutination. All right, so we have tons of effects of antibodies in action. All right, we're going to skip that one. And let's see, we're about out of time anyway. Okay, we'll probably hit these last few slides next time just because I do think they're worth going over. And then we'll start in on the respiratory chapter. All right, but otherwise, that is it for the day, folks.